Dear students, this presentation is on innate immunity, and this presentation is particularly meant for MSc Semester 3 students of Zoology. Paper code JU3024, paper name Immunology, Microbiology and Parasitology. And this presentation is for the Darrang College e-learning platform. Myself, Abhijit Das, Associate Professor, Department of Zoology, Darrang College, Tezpur. Now moving on to the first slide, the first question that comes to our mind, that what is immunity? Now basically, the immunity is a state of protection from infectious diseases. And there are two types of immunity, that is the innate immunity and the adaptive immunity. Moreover, the innate immunity is non-specific and the adaptive immunity is specific. The non-specific component, that is the innate immunity, provides the first line of defense against infection. The phagocytic cells such as macrophages and neutrophils, and the barriers such as skin and a variety of antimicrobial compounds that are synthesized by the host, all of these play an important role in innate immunity. And the major agents of adaptive immunity are lymphocytes and the antibodies and other molecules they produce. Now in this figure you can see the lines of immunity. Now in one line there is the innate immunity, and in the other line there is the adaptive immunity. Today's discussion will be focused only on innate immunity. Now innate immunity, which is also the non-specific immunity, it has got four barriers, and those barriers are anatomical barrier, then the physiological barrier, phagocytic barrier, and the inflammatory barrier. Today, now discussing about the anatomical barrier, here in this particular barrier falls the skin and the mucosal surfaces. The skin and the mucosal surfaces, they provide protective barrier against infection. It is also called as the first line of defense against infection. The skin and the surface of mucous membranes are included in this category because they are effective barriers to the entry of most microorganisms. The skin has got the sebaceous glands, and these sebaceous glands, they produce a oily substance, and that oily secretion is called as the sebum. Now the sebaceous glands, they are associated with the hair follicles. The sebaceous glands, they produce the sebum. The sebum consists of lactic acids and fatty acids, which maintain the pH of the skin between 3 and 5. And this pH, this acidic pH, in fact, it inhibits the growth of most microorganisms. However, if there is break in the skin resulting from scratches, wounds, or abrasion, then it creates the route by which infection can take place. Moreover, the skin may also be penetrated by biting insects, such as mosquitoes, mites, ticks, fleas, and sandflies, and in that process, the infection can be produced. So otherwise, the intact skin is the first line of barrier, as it prevents the entry of microorganisms into the body. In this picture, you can see the anatomy of the skin. The skin has got the epidermis. The skin has got the epidermis layer. It has got the dermis. It has got the subcutaneous layer. And in this particular dermis layer is found the sebaceous gland. And this sebaceous gland, it produces the sebum, which is oily secretion, and that gives protection against infection. Moving on to the next slide. We are also now moving on to the next barrier, in fact, and that barrier is called as a physiologic barrier. And the physiologic barrier that contribute to innate immunity include temperature, pH, and various soluble and cell-associated molecules. Now, many species are not susceptible to certain diseases simply because their normal body temperature inhibits the growth of most pathogens. For example, chicken have innate immunity to anthrax because their high body temperature inhibits the growth of bacteria. Moreover, the gastric acidity is an innate physiologic barrier to infection because very few ingested microorganisms can survive the low pH of the stomach contents. Moreover, a variety of soluble factors contribute to innate immunity. Among them are the soluble proteins such as lysozyme, interferon, and complement. Now, what is lysozyme? Lysozyme is a hydrolytic enzyme which is found in the mucous secretions and in tears. And this lysozyme, it can cleave the peptidoglycan layer of the bacterial cell wall, and thereby it degrades the bacteria and gives protection to us. Moreover, interferon comprises a group of proteins produced by virus infected cells. Among the many functions of the interferon, one of the most important function is that the interferon which is produced by virus infected cell can induce a generalized antiviral state in the nearby cells. So this picture you can see how interferon is acting as a physiological barrier. This is the virus infected cell, and this virus infected cell it produces the interferon, and this interferon bind to the interferon receptor on the nearby cell, and it induces the antiviral state in the nearby cells. Now coming on to the phagocytic barrier. This is the third barrier in fact of innate immunity. It is another important innate defense mechanism, and in the ingestion of extracellular particulate material by phagocytosis. The phagocytosis is one type of endocytosis, the general term for the uptake by a cell of materials from its environment. So phagocytic barrier is a very important barrier. Phagocytic cells, they perform this activity, and in phagocytosis, a cell's plasma membrane expands. It expands around what? It expands around the particulate material, and that particulate material, it may include the whole pathogenic microorganism, and ultimately it is engulfed in a vesicle, and that vesicle is called as the phagosome. And that phagosome ultimately binds to with the lysosome, and that lysosome has got the enzymes, and those enzymes can degrade the bacteria, and ultimately it is removed out of the cell. However, most phagocytosis is conducted by specialized cells, and those cells are such as monocyte, neutrophil, and tissue macrophages. So here in this picture, you can see how the phagocytosis is being performed. This is the bacterial cell, and this bacterial cell is uptaken by this phagocytic cell with its receptor, and this bacteria it has entered inside the phagosome. Then this phagosome it binds with the lysosome, and ultimately it forms a phagolysosome, and this leads to what? This particularly leads to a digestion of the bacteria. That means degradation of the bacteria. That is, the lysosome has got the enzymes, and those lysosomic enzymes will degrade the bacteria, and ultimately they will be removed out of the body by the process of exocytosis. and thereby it gives protection to us from different types of infection. So in this particular discussion, I have taken up three barriers, that is the anatomical barrier, the physiologic barrier, and the phagocytic barrier. In the next presentation, I shall take up the inflammatory barrier and the adaptive or specific immunity. Thank you so much.